And thanks everybody for joining. So I'm Carol Tailbridge. I'm the program director for the NINDS Translational Biomarker Program. And we also have Dr. Ram Arthachandran, who is the program director who leads the HEAL initiative and also holds the pain biomarker projects. And then Adisa C. Liu, who is the health program specialist with the teams, as well as Rakonda, who you just met. If I can actually. So, minor advancement issue here. There we go. All right, so just briefly, we're going to go over a little bit about the background about the NINDS Biomarker Program and the context relative to the rest of NINDS funding, some important terminology and concepts for the funding opportunities, and then talk a little bit more in detail about the actual funding opportunities themselves. Adisa is also going to give an overview of the NIH data sharing policy, which has actually started this year, is now in place, the new data sharing policy. And then we're going to just quickly highlight a couple of new funding opportunities that are out or are coming out. We won't talk about those in detail, but just as a FYI. All right, so clearly the mouse is going to be a problem. Let's try that again. All right, so sorry about that. So what's a biomarker? So a biomarker is a defined characteristic that can be measured as an indicator of a normal biological process, a pathogenic process, or response to an exposure or intervention, including therapeutic interventions. And this is the definition from the FDA's NIH Biomarker Working Group, the BEST glossary, which we'll talk about a little bit more as well. So at NINDS, across all of our divisions, the Division of Neuroscience, the Translational Division, where this team is, and the Division of Clinical Research, there's biomarker research that goes across all of these areas. And sort of the major differences are that within the Division of Neuroscience, which is kind of the R01 space, biomarker work focus there is more investigating mechanisms or the pathophysiology of the diseases, identifying biomarkers for generating new hypotheses about what's involved in a particular disease or condition, phenotyping, characterizing patient populations, as well as sort of the early stage technology development for detection methods for measuring biomarkers. Then the Division of Translational Research, where we are, our program is really focused on developing and evaluating biomarkers as fit for purpose tools. So this is much more product deliverable, deliverable oriented funding opportunities. And that includes analytical validation of the detection methods, and then clinical validation of the biomarkers for those specific use cases. And then finally, in our Division of Clinical Research, those programs will obviously incorporate biomarkers into many of their clinical trials. And there might also be funding opportunities where there's biomarker opportunities to leverage clinical trials or clinical networks like the Siren Network or the StrokeNet and so on. So we're going to talk about the funding opportunities in our translational program. But first, just why is it that we need more biomarkers? And you hear that all the time in all the different workshops, but what do we really mean when we say that? And this gets back to some of these different funding opportunities, but in some cases, we're talking about trying to understand the disease etiology or mechanisms. So again, like I said before, biomarkers that can help identify or generate new hypotheses about what those underlying mechanisms are for a particular disease or condition or process. And then used as tools in therapeutic development and used as tools for decision-making in clinical practice. So the R01 space is really that mechanistic space, as I said before. And then the NINDS Translational Program is focused on therapeutic biomarkers for use as therapeutic tools in clinical practice. And specifically, the goal of the program is to facilitate the validation of biomarkers and biomarker signatures as fit for purpose tools and for use in clinical trials to accelerate therapeutic development and aid in decision-making for patient care. So we have a number of funding opportunities that have been out for a while. PAR 22089 is our early entry stage R61, R33 phase mechanism for development of biomarkers or biomarker signatures. And then we have cooperative agreements, both U01 academic mechanisms and small business U44 mechanisms for analytical validation and clinical validation. And we'll talk about those more in a bit. So for all of the funding opportunities, because we're really focused on deliverable-oriented biomarker studies, it's really important to think about exactly what the use case is, the context of use, and what kind of biomarker you're actually trying to develop and validate. So the BEST glossary has provided the definitions for different types of biomarkers and how they're used in these different contexts. And that includes diagnostic, monitoring, pharmacodynamic response biomarkers, predictive, prognostic, safety, and susceptibility or risk biomarkers. And the links here are available, so when you get the slides later, you can go and read more about those. So how are biomarkers used in therapeutic development? And I say primarily because they can be used across all stages of development, but in general, pharmacodynamic response biomarkers are used more in the early stage, pre-IND or IDE phase, where there's needing to demonstrate that the biomarker or the therapeutic is actually engaging with the proposed mechanism of action, can help inform dosing studies, and help set up for successful phase 1 first-in-human studies. Then in later stage, phase 2 and phase 3, the patient stratification biomarkers, so prognostic, predictive, susceptibility, risk biomarkers, can all be very useful in helping stratify patients and reduce sample sizes needed for clinical trials, so that you're more likely to get the right therapeutic to the right patient population and be able to detect a clinically meaningful difference. Diagnostic, of course, get used across these areas where there's patients that are going to be enrolled for the therapeutic indication. And then monitoring biomarkers can be used across the whole span. Monitoring biomarkers might be validated as being really related to a particular biological process that's important for therapeutic development, and then may also be used as response biomarkers if they show a good response to the intervention and change over time. And then finally, safety biomarkers to help ensure that you don't have effects from the therapeutic that need to be identified early before a negative consequence is occurring. Now, these same definitions, when you're looking at biomarker use in clinical practice, again, the same purpose, but slightly different context of use, right? So here, with susceptibility risk biomarkers, you're really looking at patient populations that are not yet diagnosed yet, but might need additional screening or for a particular disease or condition. Then, of course, you need the biomarkers to actually diagnose the disease. And then what's the most likely prognosis? Are they more likely to have a particularly negative prognosis or positive? And how does that inform what treatment is most appropriate, which is where you get into predictive biomarkers? Are they likely to respond to one intervention versus another? And then pharmacodynamic response biomarkers, you might need to modify the therapeutic, the drug, or the dose of the therapeutic based on the patient's response. And then, of course, monitoring, again, looking at how the disease is progressing and safety for any ill effects of the treatment. So back in 2017, the FDA put out guidance for the biomarker qualification. And this is a pre-competitive space where researchers are encouraged to develop and validate biomarkers and have them qualified through the FDA as a drug development tool or a biological drug development tool. And so our program was really set up around this guidance and thinking about what kind of study designs were needed in order to help provide these types of data for biomarker qualification, or just for ensuring that they have biomarkers that are reliable within therapeutic development or clinical practice. And one of the primary things that I already mentioned before is the context of use, right? And again, this is incorporating what's the category of biomarker, and then how are you going to use that biomarker to make a decision? How is it going to inform decisions either in therapeutic development or in clinical practice? And so thinking about what's the need for the biomarker, what's the benefit, and what's the risk if the biomarker is inaccurate, and then what kind of evidence and study design is needed based on that context of use. So questions that you want to think about as you're forming and developing your context of use. What's the purpose of the biomarker? And it says drug development here, but it might be any therapeutic development or clinical practice. This is just to give you an idea of what types of things you want to think through. When's the biomarker going to be used? How is the biomarker applied? Is it going to be decisional, co-decisional? What's the context in terms of other biomarkers for that particular context of use or in clinical practice? So is it an assistive diagnostic or is it a standalone diagnostic and so on? And then, of course, what's the intended patient population? So I mentioned the biomarker definition before. We also talk about in these funding opportunities the biomarker signature. And this is just a combination of multiple variables or components to yield a patient-specific indicator. So basically multiple components of a single biomarker. And then detection method is the method that you actually use to measure the biomarker. My animation's a little off here, but a concept of interest is the aspect of the individual's clinical, biological, physical, or functional state or experience that the assessment is intended to indicate or reflect, right? So what does the biomarker, what is the concept of interest that the biomarker is supposed to reflect that's relevant for the particular disease or condition? And one that I'm sure everybody's heard of is the neurofilament light, which is a biomarker that's being used in a lot of different areas and broadly reflects the concept of axonal injury or degeneration. Neurodegeneration. And just to mention, the FDA is currently updating and thinking about the terminology around multi-component biomarkers. There was also a recent National Academies workshop last week discussing these as well. So there's a link here for more information on some of the discussions around that area. So biomarker validation process and scope. And thinking about this from sort of the early stage discovery area, which is where our first funding opportunity is set up. You're really establishing that the biomarker reflects the concept of interest, developing the optimizing the detection method to actually measure the biomarker, and then conducting initial proof of concept clinical validation to test if the biomarker can be used, is fit for purpose, can be interpreted for a particular context of use. So that might be a small scale study that's, a more focused patient population, just to demonstrate that the biomarker can be used as proposed. Analytical validation is to establish the performance characteristics of the detection method. And those include, sensitivity, specificity, accuracy, precision, and other relevant performance characteristics. Essentially, can you trust the results of the technology being used to measure the biomarker? And then finally, clinical validation is establishing that the biomarker acceptably identifies, measures, or predicts that concept of interest within a defined context of use, right? So here the focus is on the meaningfulness and how you interpret the results of the biomarker for making decisions at that individual or patient participant level. And then we don't require biomarker qualification for our funding opportunities, but the evidence from the analytical and clinical validation studies are really set up to help provide the evidence necessary for biomarker qualification if you choose to go that direction. And this is, of course, is a conclusion based on a formal regulatory process that within the stated context of use, a medical product development tool can be relied upon for the specific interpretation and application. So For the funding opportunities in our program, I'm going to talk first about the preliminary data and premise. So for the entry-level biomarker development PIR, here the evidence you really need to come in with is demonstrating that you've got supporting preliminary data or literature supporting the biological rationale for the biomarker utility in one or more contexts of use. So you can have more than one, but you need to explain how it's going to be used and what's the rationale for its use there. And then, of course, evidence that the team can conduct the proposed studies. So any preliminary data or literature supporting that as well. Preliminary data for the analytical validation. In this case, you need to already have basically done what the biomarker development PAR scope covers. So that includes demonstrating that initial clinical study might be retrospective or prospective, but demonstrating that you can use the biomarker for a particular context of use. Of course, it needs to be supported by a strong rationale for the clinical impact and the unmet need. Why is this an essential tool that's not being currently filled by another biomarker or clinical outcome assessment? And then the detection method must have already been developed. So you can have additional optimization in this PAR, but you should at least have a prototype. And then finally, in the clinical validation, this one again, adding on to these other two PARs. Now, you not only have to have demonstrated that initial proof of concept study, you also need to have the analytical validation completed for the detection method that's going to be used, because here is going to be really about demonstrating that you can get a reliable and interpretable signal within the large clinical population that is of interest. Now, in terms of the research scope, so what you can actually propose to do in each of these funding opportunities. So in the biomarker development one, this is that initial proof of concept clinical study. And again, this can be retrospective or prospective. You can include development of the detection method if that's one of the things that's needed in order to actually measure the biomarker, and that can be done in that first R61 phase. And then you can do multi-site studies here. It's not expected, but it's certainly allowed, and sometimes it's necessary for the context of use, or just in order to have the number of enrollees or data sets you need for the study. Then for analytical validation, here you might include optimization of the detection method. Multi-site validation is expected here. You can justify it. If you can justify it scientifically why it's not appropriate, that's okay, but you do need to justify it if you don't propose multi-site study. And then you can include analytical validation for the detection method. So again, you want to propose exactly what context of use or uses you're proposing to validate this for, and then evaluate the performance characteristics in terms of those, what's needed for that context of use. It also includes things like evaluating the pre-analytical variables that might impact the readout of the detection method, and establish the standard operating procedures to ensure reliability across sites and operators. And then finally, for the clinical validation PARs, this is to establish a statistical threshold needed to interpret and make decisions. So study design and statistical design is important for all of these, but it's especially critical when you're looking at the clinical validation aspect. And by the end of this, you really want to have something that's ready to be rolled out in clinical trials or in clinical practice, where you've got enough data really supporting that you can interpret it in the broad, appropriately representative patient population that it's intended for. So again, large multi-site studies are expected. These can be retrospective, but often because of concerns with pre-analytical variables and because of lack of standardization of data sets available, these often need to be prospective. And plans to qualify biomarkers with the FDA is encouraged, but it's not required. You could also go through CDRH if you're developing and trying to validate a assistive diagnostic or a device that's going to be used for a particular context of use. All right, and then just general characteristics of each of these funding opportunities. So all of these are up to 5 years in funding. In general, the analytical validation is the one that probably doesn't usually need to be 5 years. So often those are a little bit shorter. The biomarker development PAR, this is a phase mechanism. So there's milestones between the R61 and R33 phase. In the analytical and clinical validations, all the cooperative agreements, there's annual milestones that need to be included. And these need to be included in your application. They should be quantitative, very focused on what the deliverables are going to be. And they will be considered as part of your approach section by reviewers. So for all of these, the budget's not limited, but if you require more than 500,000 direct costs in any 1 year, you need to make sure that you reach out to us at least 6 weeks before the receipt date to get prior approval to come in. And you need to have a decent bit of information ready for the proposal already in order to get approval. Intellectual property, this is not a scoreable item, but it should be considered. So if you're planning on commercializing something, especially the detection method generally, you want to make sure you include your IP landscape. If you're developing an open source tool, something that is not going to be commercialized, then that's not a consideration, but you definitely want to describe that well in your data sharing plan. And then in research data and sharing plans, again, these aren't scoreable, and these are now reviewed by program. This is part of the new policy, but you need to think about how's your biomarker going to be used? How are the data that you're developing and generating for your study? What's the value of how they're shared? And think about that as you're putting together your sharing plan. And if you have questions about that as you're putting together a proposal, please reach out to program staff. All right. So again, the R61, R33 is phased. It can be 2 to 3 years for the first phase, or 2 to 3 years for the second phase, but no more than 5 years. I mentioned the budget limitations already. Applications absolutely must include both phases. Sometimes this is a point of confusion. For some investigators, you need to include both. If you don't, it'll get withdrawn. For the cooperative agreements, again, up to 5 years. Budget for the small businesses, I believe it's 3.5 million cap for phase 2. And the information on the small business budget information is at the link at the bottom there. And then what about the milestones? So these are just general types of biomarkers that should be included. So some of the ones you want to think about including are performance of the detection method, and that might be true for the R61, R33 phase, as well as the analytical validation. And demonstrate, the acceptable sensitivity, specificity, accuracy, precision, and what are those thresholds? What's the minimal acceptable threshold you need in order to demonstrate that it's going to be useful for the context of use provided. And likewise, dynamic range for the context of use. You also want to define the metrics for assessing data quality and assurance. How are you demonstrating that you're really getting the data you need? Define enrollment sample data collection requirements for the objectives of the power analysis for the study goals. Performance of the biomarker for the concept of use proposed. So now we're talking clinical validation or clinical proof of concept validation. What's the acceptable sensitivity, specificity, maybe positive predictive value or negative predictive value, accuracy, precision, and any other performance characteristics that's relevant to predict that the clinical outcome of interest is appropriate for the context of use. And then finally, progress milestones. You might include things like establishing contracts, data use agreements, material transfer agreements, and so on, or establishing standard operating procedures across sites, consulting with the FDA or submitting a letter of intent to the biomarker qualification is a good one if you're in the analytical or clinical validation PARs. You can do it in the development PAR as well. It's just usually those are a bit too early for that. So those are just some examples of the types of biomarkers that should be included or could be included. And then finally, studies that are not in scope. So if the primary intent is to develop therapeutic agent or devices, those would not be out of scope. Those would not be in scope. We've got other funding programs in Division of Translational Research and NINDS in general to help with those. Evaluation of clinical efficacy, effectiveness, or clinical management. Again, most of that's in our Division of Clinical Research. Natural history studies without specifically planning on validating a biomarker for a particular concept of use is out of scope. We no longer have work with animal models in any of these PARs, so those are all out of scope. And then if you don't include milestones in your applications, those get withdrawn as well. And of course, it needs to be within the mission of NINDS. NIA is also signed on to our PAR 22089, which is the development PAR. So we put this up front just to make sure that nobody wastes their time submitting something that's not going to be able to come in for review. So finally, we're talking about the funding mechanism. So for those of you who have been funded through NINDS before, we're primarily a pay-line driven institute, which means that we set a funding opportunity, or we set a funding pay line for our R1s and R21s each year based on the budget. I think right now it's around 14 percentile. But for these translational programs, there's a pay plan that's developed. And so essentially we look at the top scoring applications for any given round. It's usually the top 20-25%. And we'll ask you to respond to the summary statement critiques if you are within that top scoring range. And then those all get discussed with leadership, and a final pay plan that's usually around the same as the pay line will be determined. And so, the higher priority will be those that address a really clear unmet medical need. So in terms of reviewers, aspects that they're reviewing and thinking about, that would be significance, be supported by a strong biological rationale. So that would be under premise and significance, including a carefully designed and of course rigorous plan for performance evaluation. So that's the approach and the milestones, including a plan for standardization of data collection and samples for use in the validation. Again, so that's approach item. And then providing strong justification for the utility of the biomarker in the clinical setting or the added value in the clinical research design. So both significance and innovation aspects there. And then that you, of course, have the expertise and resources needed for the feasibility of the studies being proposed. So that would include items under investigators, available resources and approach when you're looking at the review items. And as I mentioned, all investigators within that top scoring range are asked to respond to the summary statement critiques. All right, and if Adisa is ready, I'm going to hand it over to her, and she's going to talk a little bit more about the data sharing and management policy. Adisa, are you ready?
Yeah, thank you, Carol. Thank you, Carol. So the NIH recently, basically at the beginning of this year, issued a data management and sharing policy in order to promote sharing of scientific data. And obviously in the name of science, that will basically facilitate, in a sense, the development of treatments and products for the improvement of human health. So the goal of this data sharing policy is to basically have investigators kind of plan for the managing and the sharing of the scientific data. Obviously submit a plan that has to be reviewed and approved by the NIH program staff, as well as kind of comply to basically the approved plan. It is important to note that the plans are not part of the scored peer review criteria, unless it is kind of specifically stated in the notice of funding opportunity. Next slide, please. Thank you. And when we basically talk about any scientific data, that includes anything from omics data, imaging data, biological data, and phenotype data. But when we're in a sense focalizing on foreign collaborations, it's and especially with investigators that are that have foreign institution or that are collecting data from foreign institution, it is important that you do kind of are familiar with their policies when it comes to governing data in the different countries, and address these aspects as part of the applications, because that will be reviewed by the NIH program staff, and that will be compliant and implemented throughout the project. And when it comes to kind of proprietary data, we basically do note, and we do basically let investigators know that for any SBIR and any small business, transfer technology awardees, they can withhold the applicable data for about 20 years after the award date. And NIH does understand that, for these particular awardees. Next slide, please. Thank you. And when it comes to basically model organisms and genomic data sharing, it is important to, in a sense, make sure that is stated in the application or the proposal, and is also highly of importance to basically justify and show any justification as to why data it's not going to be shared, or it's restricted, or if it's not possible. And as soon as, in a sense, the project or the end of the performance period comes to an end, it is important to also, have the scientific data to be made accessible as soon as that particular period comes to an end. Next slide, please. And you can also find more information about, the planning and the budgeting of the data management, as well as, the sharing of the scientific data repositories and the data management on our NIH website at the sharing.nih.gov. Thank you.
Thanks, Adisa. All right. Finally, there's a number of resources that are available that you should be aware of. So NINDS's Human Biomarkers Biospecimen Data Repository or BioSend. So if you're collecting new samples, we highly encourage you to bank your samples, extra samples with BioSend. And you could also use this and submit a request to access those samples. And links are here to tell you more about what biosamples are available, as well as the NIH Neurobiobank, Human Cell and Data Repository, the Accelerated Medicine Partnership for Parkinson's, and the Federal Interagency Traumatic Brain Injury Research, or FITBR. And then I should say the link here for NIH data sharing repositories. So you can look at what types of repositories are available to share your data when you're thinking about your data sharing plan as well. All right, so in the last couple of minutes here, I'm just going to point out a couple of funding opportunities. I'm not going to talk about these, but there's an ADRD, so the Alzheimer's disease related dementias funding opportunity. They put out new funding opportunities each year. This is a RFA, so this is a set aside funding for an application for an award for a U19 grant. These are a large program grant that includes administrative core, data management and statistics core, clinical core, and 2 to 4 research projects. And these are specifically for differential diagnosis of 2 or more Alzheimer's disease related dementias that present similarly clinically. And these are all required to work on clinical validation and qualification through FDA's Biomarker Qualification Program. So if you're interested in learning more, please reach out to me. And then there is a reissue of one of ROM's funding opportunities for the HEAL program, which is discovery of biomarkers and biomarker signatures to facilitate clinical trials for pain therapeutics. And these are specifically for developing and validating biomarkers for response monitoring or predicting response to therapeutics. So that is out now, and please reach out to Ron if you have more questions for him. And then this is brand new, as the concept's been approved, but the funding opportunity is not out yet. So ROM has one on developing remote or patient wearable devices, derived objective biosignatures or functional assessments to monitor pain. So focused on monitoring here. And again, please reach out to Ron if you have questions. And last, there's a new notice of special interest, just encouraging applicants to advance research for ALS. And that includes our development PAR is on that NOCI, as is the small business parent grants. So biomarker work can be done in either of those. So I encourage you to list this notice on your application, which highlights for NINDS staff that you're responding in this notice as an ALS grant. And with that, I'm going to pull off the slides and we can answer any questions.
Thanks, Carol. Again, for those that may have joined late, please submit your questions in the chat section below and we will answer them. Please remain muted. Thank you.
And thanks. I see Chris Treating Swanson Fisher is on. I just sent a link to the BioSign too. Thanks, Chris.
Yeah, no worries. Hi, everybody. I'm Chris Swanson Fisher. I'm a program officer currently for the BioSign and NHCDR repository. So if you have any questions about leveraging these repositories, either the existing biospecimens or cell lines, or interested in banking with those affiliated repositories, please reach out to me. I'm happy to assist and walk you through the process. Thanks.
Yes, and the slides will be available. Those will go out in a couple of days after the webinar is over. And I also see there's a direct question to me, and I would ask that person to reach out and we can set up a call. You can either email Rakonda or me directly, and we can just set up a time. We can talk about that a little bit more in detail. Let's see. So June Lee, to answer your question, for biomarker development, are in vitro and in vivo data both needed? It depends on the context of use, what exactly you're trying to do. You know, if I don't know if you want to unmute and add a little bit more detail. I mean, in general, like every study section, every review section, it's hard to say exactly what preliminary data you need. You know, do investigators believe there's good premise? Do they believe you can do the work? That type of thing. So I think it kind of depends on exactly what you're proposing to do. But yeah, go ahead.
Sure, thank you. So I'm asking because now for example, we're using these patient-specific stem cell models from these genetic disease patients. So we can using to look at for the biomarker development, look at what the changes, metabolism changes. I'm wondering, is this when in the biomarker development grant, at the beginning, do we also need to, in addition to this human iPS stem cell model, do we also need to validate that in animal models?
Not necessarily. I mean, I would say, yeah, it's not a requirement for the funding opportunity. That's the short answer, I guess. Okay. So you would include what data you have. In general, we encourage work that's from previous human data as much as possible. So that's the most important thing.
Okay, sure. Thank you.
Mm-hmm. Other questions? And I know we do these a couple of times a year, so these are online. We don't have a big group today, but if there's any specific questions or if anybody wants to set up a call to discuss a project, please just let me know. We'll give it maybe another minute for anybody who's still thinking about their questions, and otherwise we'll give you all a little bit of time back.
I do have another question. So you mentioned that the last there is a funding opportunity for this neuromuscular related disease and also ARS. So now like there are also a lot of this kind of like rather rare neuromuscular disease, for example, hereditary spotty paraplegia. I just wondering, is that also in that RFA considered?
Can you repeat that last part of the question? Sorry.
Sure, certainly. So there are some rare diseases that also affect this neuromuscular, like particular, like this is like a nerve degeneration for this cells. Like it's called hereditary spotty paraplegia, a disease.
Oh, I see. And is that eligible for the?
Is that eligible for this?
Yeah, no, that wouldn't be. There are some rare disease specific funding opportunities, including the clinical trial readiness funding opportunity. But the notice, and I should say that the notice doesn't have special set aside funding with it. It's just really flagging ALS, which we just had a strategic planning process from, and so
okay,
highlighting those projects. Yeah.
Okay,
Thanks for the
sure.
question.
Sure, thanks.
Thanks, Chris.
Oh, thank you.
Yeah, of course. And I can certainly follow up with you offline as well, June, with a number of our other active funding opportunity announcements, not just with NINDS, but through some of our other institutes, including support for HSP research as well, too. So I'll happy to share that with you.
Okay, great. Thank you.
Of course.
All right, last call. Any questions? All right, great. Thank you all so much for joining, and thanks to the team for all the extra input. And please let us know if you have any follow-up questions after the slides go out or later in the day. Take care, everybody.
Thank you.